Hi all. So friends, in the last class we discussed about the concept of nitrogen metabolism and we completed. From today we are going to start discussing about the plant hormones. So friends, we know in our CSIR exam as well as IIT JAM exam and GATE XL exam also plant hormone, even in the GATE BT also plant hormones are the one of the most important topic where we use it to get the questions more often. We can get the question from the signaling also, we can get the question from structural related and composition also. Structural related, you don't need to remember the structure, actually you have to remember the composition. And you should also remember the site of synthesis, right? So let's try to understand one by one harmony elaboratively. So I'll explain from basic to advanced level. You can understand everything very easily. So first of all, listen, what are plant hormones? How can we define the word plant hormones? Friends, plant hormones are chemical messenger. What do you mean by that? They are acting like a chemical messenger. They are involved in passing the signal from one area to the another area. That's why we can call plant hormones are chemical messenger. Even plant hormones plays a crucial role in the signal transduction pathway, that is carrying the signal from one place to the till nucleus level, to be specific. And plant hormones are do not have a nutritious value, that's why we can call plant hormones are non-nutritious. And very important point, the site of production and the site of action is entirely different. What do you mean by that? Friends, in the plant body, suppose let's take an example of auxin. Where auxin is present, auxin is synthesized in the shoot. But friends, we all commonly know that where the auxin is going to show their action in the root. Yes or no? Yes. Now if you think about the cytokinin, where does the cytokinin is basically produced? Cytokinin synthesized in the root. But where does the cytokinin is going to show their action in the shoot? Is it? Auxin can show the action on the root but produced in the shoot. Cytokinin show its action in the shoot but produced in the root. Don't confuse with that. That's why we can say site of production and the site of action is entirely different. Now, another very important point. All the plant hormones produced in very low concentration. What do you mean by that, friends? Which means low the hormone concentration, high the activity. High the concentration, low the activity. So that's why if a plant hormone want to show their action in the plant body, they should be always synthesized in very low concentration. Please kindly do not forget that particular point at all. Friends, every one of you listen. How did we classified the plants in the plant body? Actually, friends, based on the action, means whether the plant hormone is promoting the growth or the plant hormone is inhibiting the growth. So based on their promoting and inhibiting the growth, we categorized into two major types. What are those? One we called, friends, a growth promoters. What is the first category? The first category is the growth promoters, and second category we call it as, friends, growth inhibitors. Very simple point. One we called growth promoter and another one we called, friends, growth inhibitors. Just keep that in your mind. Okay, now growth promoter, name itself indicating, right, is going to promote the growth. What are the example? Auxin, cytokinin, gibberellic acid. And we all know, friends, one and only the growth inhibitor, that is the abscisic acid. So auxin, cytokinin, gibberellic acids are acting like a growth promoter, abscisic acid acting like a growth inhibitor. Now my question is very clear. What is that? Then what about the ethylene? So friends, actually ethylene can act like both depending on the condition. Ethylene act like a growth promoter also as well as the growth inhibitor also. My friends, these five are the major plant hormones abundantly found in every single plant. Apart from the five major plant hormone, there are two other hormones are there. What are those? We called jasmonic acid and friends, followed by the brassinosteroids. Please kindly do not forget these two also. What is the speciality of the jasmonic acid and brassinosteroid? That's what I will tell you in the upcoming slide. They also can act like a growth promoter also, even growth inhibitor also, depending on the condition. Let's understand one by one. So let's start with the first and most important hormone, that is the auxin. So friends, I'll explain elaboratively, just pay attention. So first point I want every one of you to remember is that auxin is the major plant hormone involved in the cell division also, cell elongation also. Friends, the moment you heard the name of auxin, I want every one of you commonly remember one particular point which we called auxin is a rooting hormone. This is the first point I want you people to remember. The moment you heard the name of auxin, you have to just remember as a rooting hormone. Before understanding depth of the auxin, let's try to understand little bit about discovery of the auxin. Friends, I will explain elaboratively, just pay attention and ask me if you have any doubts. Please comment. In the year 1860s, two intelligent scientists who Charles Darwin, first listen to me completely, you will get a clear cut idea. Charles Darwin and Francis Darwin. What these two people did, they started studying coleoptile. This is called coleoptile. Now you can ask me a question. Sir, first of all, what is a coleoptile? Friends, everyone see, this is the plant, right? So this is the shoot. Friends, this particular tip, the tip of the shoot, this is called tip. The tip of the shoot only we called coleoptile. What Darwin and his son does, they have taken the coleoptile. Up after taking the coleoptile, they have exposed it to the unilateral sunlight. My friends, what do you mean by the word unilateral sunlight? Unilateral sunlight means the movement of the sunlight, it is in one direction. This is called unidirectional. Upon exposing the coleoptile to the unilateral direction, they observed that there is a bend. There is a bend in the coleoptile. That is what he observed. But then Darwin and his son Francis Darwin concluded. What they concluded? They concluded that yes, there is a chemical. Everyone listen my words. There is a chemical present in the tip of the coleoptile. There is a chemical present in the tip of the coleoptile. Because of this particular chemical only, this chemical, when absorb the light, it moves from lighter region to the darker region, which leads to the bending of coleoptile. What friends? This chemical comes from light to the dark direction and it will allow the coleoptile to bend. Always keep that in your mind. But unfortunately, Darwin and his son, they did not get any credit for this. Later, a very intelligent scientist, I want you people to remember. We can expect a simple questions from the experiment, but what mostly we won't get, but we should not take the chance. Okay. Francis went. What he has done, same thing. He has taken the coleoptile. Friends, listen my statements carefully. He has taken the coleoptile. From where he has taken the coleoptile? He has taken the coleoptile from the Avena grass seedlings. Friends, what are Avena grass seedlings? Nothing but a common oat plant. He has taken the coleoptile from the common oat plant. First, initially, he has exposed to the unilateral sunlight. When he exposed to the unilateral sunlight, it starts bending. The coleoptile starts bending upon perceiving the sunlight. Once again, what he has done, he has taken the coleoptile. Friends, you know this time what he has done, he cut the tip. See, look into this. He just cut the tip. Whatever the tip he just got cut, this is the tip, right? He just kept in the agar solution. We all know, right? If you keep something in agar, it will start diffusing it immediately. We all commonly know about it. What happened now without a tip? How does it look like? It looks like this. We called it as what? A decapitated part, means where there is a no presence of the tip or a cap. Now my friends, you know what happened? Without a tip, he started exposing to the unilateral sunlight. My friends, surprisingly, it was not at all bent. It was not bent. Now later, once again, what happened? He has taken the same thing, and whatever the tip he kept on the agar plate or agar medium, he has taken that tip back and he placed on the decapitated part. And once again, immediately he started exposing to this, exposing to the unilateral sunlight. My friends, immediately it started showing bending. So friends, what you have to understand by the simple experiment, you have to understand that yes, there is a chemical exclusively present in the tip of the coleoptile only. Upon perceiving the light, it starts moving from the lighter region to the darker region and allowing the coleoptile to bend. How? By the process of cell elongation. Let me summarize this. What he has done: first he has taken the avena grass seedling, he exposed it to the unilateral sunlight, it starts showing bending. So now this time what he has done, he cut the tip. Whatever the tip he cut, he just kept in the agar medium. What without a tip, he started exposing to the unilateral sunlight, it's not at all bending. Now whatever the tip he kept in the agar, he just kept back, and then he started exposing to the sunlight. My friend, surprisingly it starts bending. Then he understand, yes, there is a chemical exclusively present only in the tip of the coleoptile. Francis Siswent is the scientist who isolated that chemical from the tip of the coleoptile. He is the one who has given the name auxin. He is the one who has given the name auxin, which means to grow. The word, the meaning of auxin is to grow. Never forget. Later, another scientist named as Kogel, you know what he has done? He has isolated the auxin from the human urine. Even he has isolated auxin from the patients which are suffering from pellagra, means the patients who are deficient of vitamin niacin. In their urine also they have identified a traces of the auxin. But friends, surprisingly, till today we did not clearly understood what is the role of hormone auxin in the human urine. Till now we did not understand this. Okay, this is the experiment of Darwin, right? And this is the experiment of Boyson and Jensen, and this is the experiment of Wendt. You know what the Boyson and Jensen does? Same. He has taken the coleoptile. Now you see here one side of the coleoptile. Which side of the coleoptile? The coleoptile to which we are passing the light. See, this is the unilateral direction. My friends, just near that particular unilateral direction on the tip, we kept a mica piece. Mica piece means, friends, a glass piece. Then coleoptile is bending very easily. There's no doubt about it. But next time what Boyson and Jensen does, this is the direction where the light is passing. On opposite direction, they kept the mica piece. That time, friends, the coleoptile is not bending. Why? Because here you only try to understand when the auxin is moving from one area lighter to the darker region, what happening? Mica piece is not allowing this particular auxin. As a result, what happen? Auxin will fail in elongating the cell. When the cell was not at all elongating, my friends, how does the coleoptile will bend? So coleoptile cannot bend. So what is the conclusion I want you to know? See, this is how exactly coleoptile looks like. Whenever coleoptile exposes to the unilateral sunlight, the auxin present in the lighter region start moving to the darker region and start start elongating the cell, which leads to bending of the coleoptile. Please kindly don't forget that particular point at all. Now we understand a little bit about the discovery. Okay, we don't get that, but we have to know it. Now this is important for the exam. What are the different types of the auxin? Friends, there are different type of auxins are there. Everyone listen. Natural auxin, synthetic auxin, free auxin, then friends, followed by the bound auxin. Let me tell you, what do you mean by the word natural auxin? Name itself indicating na natural auxin, the auxins which are synthesized naturally in the plant body, that is what do we call natural auxin. Example, IAA, indole acetic acid, IBA, indole butyric acid. A simple point. Okay, but friends, I want every one of you remember one more common point. How many number of auxins may present in the plant body? The most common natural form of auxin. Once again, listen to me. The most common natural form of the auxin, it is always IAA only, indole acetic acid only. Please kindly do not forget this statement also. Next, synthetic auxin. What do you mean by the word synthetic auxin? Name itself indicating na, which is synthesized artificially, man-made. Naphthalene acetic acid, NPA, and very important one which was asked for the exam, that is 2,4 dichloro phenoxyacetic acid. Never ever forget that. Next one, 2,4,5 trichlorophenoxyacetic acid. Such a simple to remember. Friends, 2,4 are the carbon number where the chlorofunctional groups are present. 2,4,5 are the carbon number where the chlorofunctional groups are present. My friends, the point I want you people to remember for the exam, and which was asked in the exam also, that is what, friends? These two are basically act like VD sites. Please kindly do not forget this particular statement under any circumstances. Now, friends, don't get confused here. What are the differences between the free auxin, bound auxin? Free auxin means, listen my statements carefully. Free auxin means, free auxin, it may be synthetic also. A free auxin may be natural also. But the point you have to remember is that if you want to isolate a free auxin, if you are not using any organic solvent, listen, if you want to isolate any auxin, if you are not using any kind of organic solvent, that auxins are called as free auxin. Means the auxin can be synthesized without organic solvent, which can be isolated without organic solvents like ethanol, ether, chloroform, carbon tetrachloride, all this. Those auxins called free auxin. A free auxin may be natural also, a free auxin may be synthetic also. But if I want to isolate a auxin, if I started using any organic solvent, then I call this particular auxins as a bound auxin. My friends, please kindly don't forget this particular statement under any circumstances. Now, site of synthesis. Everyone pay attention. Shoot apical meristems are the major site of synthesis of auxin. I can say 100% auxin can be biosynthesized at the shoot apical meristem. But friends, even at a lower concentration, at very low concentration, every part of the plant body consists of this particular auxin at low concentration. At very low concentration, every part of the plant body consists of this particular auxin. Please kindly don't forget that particular point. Okay, now once again the question was asked in the exam, which of the following can act like a precursor? for the biosynthesis of auxin. I want every one of you remember, friends, the auxin actually what derivative? It is a indole derivative. You are able to see here. It's a indole derivative. Now friends, this particular auxin can be biosynthesized with the help of an important amino acid, which is what? A tryptophan. Tryptophan. We are all commonly know, right friends? Tryptophan specialty is what? Tryptophan can act like a aromatic amino acid. So always keep that in your mind. It's an aromatic amino acid. Okay. So how auxin can be biosynthesized? Friends, auxin can be biosynthesized by using different pathways. I'll tell you. One we called tryptophan dependent pathway. First one, tryptophan dependent pathway. Second one, my friends, we will be calling it as tryptophan independent pathway. Never ever forget this particular point also. It's a dependent pathway, then followed by, friends, independent pathway. Never forget. Now you can ask me, sir, which pathway will occur more prominently in the plant body? Undoubtedly, friends, tryptophan dependent pathway. You can ask me, first of all, sir, what do you mean by the word dependent and independent pathways? Dependent pathway means the name itself indicating, now friends, tryptophan can acting like a starting precursor molecule. Once again, understand, tryptophan acting like a starting precursor molecule. That's why we call tryptophan dependent pathway. Now tryptophan independent pathway, tryptophan is not involved directly. Some other molecules will be involved. Tryptophan will be involved in the middle. That's why we call tryptophan independent pathway. But the point every one of you has to remember, plant can undergo which pathway? Mostly tryptophan independent pathway only. Friends, there are many tryptophan independent pathways are there, friends. One we call it as IPA pathway, indole pyruvic acid. One we called IAN pathway, indole acetonitrile pathway. Please kindly don't forget, indole acetonitrile pathway. Don't forget. Then another one, you don't need to remember IAN and IAM pathways, but I want you to majorly remember IPA pathway, indole acetamide. This is acetonitrile pathway, acetamide pathway, major pathway. So once again, for the exam point of view, you have to remember the major principal plant pathway used is always IPA, indole 3 pyruvic acid pathway only. Never ever forget that particular point. Tryptophan. So everyone listen my words carefully. First we are going to discuss about the IPA pathway. Friends, you people no need to remember even a single structure also. Single structure also you no need to remember. What you people has to remember for the exam point of view is that the schematic presentation, which means which is the first step, which is the second step. That's the thing you have to remember. You no need to concentrate on the structure at all. Indole 3 pyruvic acid pathway. So what is the first one? Tryptophan. So friends, tryptophan can be treated by an important enzyme. What is the name of the enzyme by which a tryptophan can be treated? We call, my friends, tryptophan transaminase enzyme. Please kindly don't forget this statement at all. Tryptophan transaminase enzyme. What is the speciality of this particular transaminase enzyme? Name itself indicating it transfer the amine group. Everyone look into this. Such a simple structure it is. Tryptophan can be treated by an enzyme tryptophan transaminase which remove the amino group. As a result, the tryptophan can be converted into an intermediate product which we called indole 3 pyruvic acid. Always keep that in your mind. Indole 3 pyruvic acid. So every one of you try to understand what I am talking here. The stable or intermediate product produced is the IPA. That's why the name of the pathway also given as IPA pathway. Simple step. Now, what is the fate of this particular indole 3-pyruvic acid? This particular indole 3-pyruvic acid can be treated. Treated by whom? It can be treated by an important enzyme. What do we call it as? IPA decarboxylase enzyme. Now friends, here once again, if you recall, transaminase enzyme remove the amino group, decarboxylase enzyme remove the carboxyl group. Then upon removal of the carboxyl group, indole 3-pyruvic acid converted into indole 3-acetaldehyde. Now, indole 3-acetaldehyde, upon treated by an enzyme, indole 3-acetaldehyde dehydrogenase, ultimately it is going to give rise to indole acetic acid. Please kindly do not forget this particular statement at all. Once again, let me quickly summarize you. What is IPA pathway? Tryptophan can be treated by an enzyme called tryptophan transaminase. Then it is converted into IPA, which is a stable and intermediate compound. That's why name of the pathway is IPA pathway. Then IPA can be converted into indole 3-acetaldehyde upon treating with the IPA decarboxylase enzyme. Now this indole 3-acetaldehyde can be converted into what, friends? Indole 3-acetic acid. Simple one. Now this is IPA pathway. Is there any necessity to remember IAN? And is there any necessity to remember IAM? And is there any necessity to remember TAM pathways? Not at all. You no need to remember, and because they are not most significant pathways in the plant body. Hence, it's not at all necessary to remember all these. What you have to focus is majorly IPA pathway only. Okay, now let's understand the most important where we use it to get the questions more often, that is functions. Let's understand one by one. What are the major functions performed by the auxin? Let's understand the first and unique function. It stimulates rooting, that is root initiation and stem cutting and lateral root development. So friends, everyone try to listen to me what exactly I'm talking here. It stimulates rooting. I hope if you quickly recall my words, what I have told to you. Auxin is a rooting hormone, yes or no? Yes. Auxin is synthesized where? Auxin is synthesized in the shoot, correct na? From the shoot, auxin travels to the root. Once auxin travels to the root, it causes the root to become elongated, which we called root elongation or lateral root development. Primary root, secondary root, tertiary root, terminal roots. These are all can be formed by what, friends? Auxin. That's the first point. Anyway, we will explain one by one elaboratively. It stimulates rooting. Second one, it stimulates cell elongation and, my friends, cell division. Let me explain you one more time elaboratively. Cell elongation and cell division. I'll tell you how exactly it elongate the cell by using this particular picture. Okay, before that, let me explain you the overview. This point we have to read very elaboratively, then only we can understand. Okay, friends, the most important point, auxin promote apical dominance. This is one of the most important point, friends. It promotes apical dominance. Friends, what is this promoting the apical dominance? Everyone listen carefully. Friends, very important point it is. Imagine this is the shoot. Here what is present? Auxin is present. Friends, whenever auxin is present in the tip of the shoot, you know what auxin will do? Auxin never ever yellow. What it never ever allow? It never ever allow the development of the lateral roots. Lateral branches. This point is very important. Until and unless auxin is present in the tip of the plant or the tip of the shoot, it never allow the development of a lateral branches. But friends, one unique point every one of you has to remember, friends, the auxin action can be suppressed by what? Cytokinin. So what cytokinin will do? Cytokinin will dominate the auxin. Then the cytokinin will yellow the lateral root development. So friends, what is the point? All of you has to remember, auxin promoting the apical dominance, so it never ever allow the lateral branches to develop. But the cytokinin suppress the level of auxin and it will allow the development of the lateral branches. So my friends, what's the common point I can say? I can say here a very important point, that is auxin and cytokinin. Friends, both are antagonistic to each other. This is such an important point it is. Auxin and cytokinin both are antagonistic to each other, means which are acting oppositely, which we call antagonism. So that's the most important step, that is the promoting apical dominance. Now auxin prevents, see, such an important step it is also. Auxin prevents abscission of young leaf and young fruit. Friends, you only try to understand, do you think young leaf and young fruit will they fall, or will they fall down on their own? No, until and unless there is a hormonal balance or any disease or condition, then only they fall. You know what is the speciality of auxin? When the auxin is present in the plant body, auxin never ever yellow the young leaf and young fruit to fall down. Always keep that in your point, in your mind. Next one, auxin, see the interesting point. Auxin only stimulate the abscission of the old leaf and old fruit. What do you mean by that? Which means auxin only preventing young leaf and young fruit not to fall. But auxin only stimulate when once the leaf become mature, it become older, it has to fall down. Then only the new one will come. That process also can be mediated by auxin only. Auxin stimulate abscission of old leaf and old fruit. Please kindly don't forget this statement. Next one, auxin induces parthenocarpy. This point also we're gonna discuss elaboratively in tomatoes and strawberry. So parthenocarpy is what, friends? Seedless fruits without fertilization. Now wait, not nowadays. From the long back only we are going to get banana seedless, grapes seedless, apple seedless. These are all what? Parthenocarpy, right? This can be induced by a hormone auxin. And don't confuse with this point. This is we already read. It promote flowering as well as a femaleness in dioecious flower. Dioecious means people will think that it's a bisexual. No, dioecious means either male or female, either male or female. That's what means dioecious. So dioecious means monosexual. Bias, understand? Monoecious means bisexual. Here what is the point? Auxin promote the flowering as well as femaleness in dioecious flower, means in a monosexual plant the auxin will helpful in the development of female characteristics when compared to the male characteristics. That's the point I want you people to remember without fail. Okay, so friends, that's all for now. In the next part we will discuss all these points one by one elaboratively and including the signaling. After this we will start with the another hormone, abscisic acid. So in the next class we will finish the auxin signaling and we will move to the abscisic acid. Okay, thank you all friends, thank you all.